she'll be putting it in throughout the meeting, a little bit in the middle and definitely toward the end. But when the webinar closes, that link goes away. So you'll need to catch it before we formally end the webinar. All right. So now it gives me great pleasure to introduce Dr. Delaney Fisher. And Dr. Fisher completed her graduate training in biomedical engineering in the focused ultrasound lab of Dr. Richard Price, who is at the University of Virginia. Currently, she's a postdoc at Duke University in the gene therapy lab of Dr. Aravind Asokan. He is a close CCM collaborator of Doug Marchuk, as well as a gene therapy researcher. Doug Marchuk is the chair of the Alliance to Cure Scientific Advisory Board. Delaney, now Dr. Fisher. It is wonderful to have you here. I have enjoyed every talk that you've given at our patient meetings, and it's great to be able to have you join us here on a webinar for the whole nation or world actually to see. So without further ado, I will let you get started. With a little further ado. She has shared with me that she has about 45 minutes of material to cover. So it's quite a bit because she has a lot to say. After she's done, we'll take the questions then. We'll have about 15 minutes. I will be curating them. If we don't get to your question in the time of the webinar, we'll try to follow up with you afterwards. All right, it's all yours.
All right, thank you so much, Connie, for the introduction, and I'm really excited to be here tonight. Thank you all for joining. If you've been following along, you'll get to see some new updates tonight. And if you're new, no worries. We're going to go over the basics of focused ultrasound as well. So hopefully we'll cover a wide spread here. So as Connie mentioned, I did recently graduate from the University of Virginia, and I thought I might just give a little bit of my journey with CCM as a little intro to how I got started in this space. So a quick little timeline. I started at the University of Virginia and joined the Ph.D. program there August 2018. I joined the Price Lab at 2018 in December. And then in August of 2020, Peter Therapeutic, who was also at the University of Virginia, came to us with this CCM collaboration. He had just created this mouse model of CCM, and he thought that we would be a great team to start exploring focused ultrasound for this disorder, and we agreed. So we started this project with him. In November of 2021, we actually treated our first mouse with focused ultrasound, and in the time in between, we were characterizing this model with MRI so that we could understand how to best treat it with focused ultrasound. And then in December of 2022, we got another graduate student, Tanya Cruz, to join the Price Lab, and she's actually continuing this work now in my absence. And she's also expanding that to computational models as well, where she can better understand drug delivery for CCM and good drug candidates. And then in December, I graduated and defended my PhD, and we were also thankfully awarded an R01 from the NIH. So we received this $3.1 million grant to continue this work for focused ultrasound research in the CCM space. And then, as Connie said, I recently started my postdoc at Duke University, where I'm working with Aravind Asokan and Doug Marchuk to continue in the CCM space and work on gene therapy. But tonight we'll be focusing on a focused ultrasound, and specifically we'll start off this first part of the talk with what is focused ultrasound, how does it work, how is it being used for other diseases to kind of get the broad landscape. And then we'll zoom in on how focused ultrasound can be used for treating cavernomas. And so we'll start by looking first at blood-brain barrier opening in itself, how that works, and then also combining this approach to enhanced drug delivery. So starting off, focused ultrasound, what is it? So it is a rapidly evolving, non-invasive therapeutic technology that has the potential to treat many different medical disorders beyond just CCM, using ultrasound energies that we can target deep within the body without any incisions or radiation. And specifically, this therapy combines the use of two technologies. So one is focused ultrasound itself, which is what provides the sound energy to treat deep within the body precisely and non-invasively. And then we usually couple it with some form of imaging, and for the brain, that's typically going to be MRI. So that allows us to identify the target tissue and also to confirm the effectiveness of this treatment afterwards. And then to help frame what focused ultrasound is, I think it's really helpful to kind of put it in the context of thinking about diagnostic ultrasound, which most of us are familiar with as this way to monitor things inside of the body like pregnancies. And with focused ultrasound, we're essentially just taking these same ultrasound waves and concentrating them into a very small volume, where we can then produce a wide range of mechanical or thermal effects to the target tissue. And so I like to use this analogy of thinking of a magnifying glass and holding that up to the sun and concentrating these sunbeams such that we can actually cause heating of a leaf. In the same way with focused ultrasound, we're really just concentrating sound waves in this case so that we can then concentrate and intensify that energy to produce a therapeutic effect. And then I also just want to remind everyone in the audience that sound waves are really just pressure waves. So that means that during a sound wave, we're basically just oscillating between these states of high and low pressure over time. And then we can manipulate these sound waves in many different ways, such as increasing or decreasing the pressure or the amplitude of these sound waves, and continuously applying the sound wave over time, or breaking it up into short pulses for short periods of time. And these manipulations of the sound wave is what can result in many different types of mechanical and thermal effects, which then allows us to provide a wide variety of biological effects within the tissue. So changing the way that the sound wave is generated causes different biological and therapeutic effects for focused ultrasound. And to give a few examples here, so for instance, if we apply a very high pressure at short frequencies, like this in yellow, we can induce 9 different biological effects in the tissue, one of which is breaking up blood clots. If we apply a more moderate pressure over a short period of time, that can be used to produce 12 different biological effects, one of which is eliciting this immune system response. If we apply a low pressure ultrasound wave for a short period of time, that allows us to produce 27 different biological effects, one of which is drug delivery, also is blood-brain barrier opening. And then finally, if we apply this continuous sound wave, as seen in purple, over a long period of time, that allows us to create a thermal effect and 10 specific biological effects from that, one of which being tumor destruction. So as you can see, by simply changing the sound waves, we get all these different responses that we can use to create different therapeutic effects and treat different disorders. And so all of these biological effects that we can produce with focused ultrasound is what makes it so versatile for treating many different diseases. So if we look at this diagram, we can see that focused ultrasound is used to treat a lot of different disorders, ranging from women's health issues to pulmonary issues and so much more, and of course neurological, as we'll be speaking of tonight. And with all of these different diseases that focused ultrasound is being used for, the development of focused ultrasound treatment ranges from these early preclinical stages where everything is still happening in the lab, and then it can range all the way to FDA approval and insurance reimbursement. And so some examples here are brain metastases from melanoma are still in these preclinical phases. So when we look at bone metastases, that's actually already FDA approved and reimbursed. And this is just a very small subset that I'm showing you in the slide. There's actually over 150 different disorders that are being pursued as of 2021 with focused ultrasound. And so far, patients have undergone over 565,000 treatments with focused ultrasound. And many of these have centered on treatment for different tumors, but there's also been a rise in patients that are being treated with neurological disorders with focused ultrasound. Many of those will be essential tremor, Parkinson's disease, and Alzheimer's disease patients. And so with all of this in mind, I want to begin to explore how focused ultrasound could be used to treat cavernomas. And again, we'll be talking about initially this temporary blood-brain barrier opening approach, and then we'll dive into how it can also be used to enhance drug delivery. And so as I'm sure everyone in this audience is more than aware, the current standard of care for CCM is surgical resection of the cavernoma. But of course, this isn't always going to be the best option for all patients. And many times there are lesions that are not really good options for surgical resection due to their location. And that really motivates the need for developing these non-invasive approaches like focused ultrasound, which can target specific cavernomas and reach even these deep-seated lesions in the brain. And while the motivation for treatment is high, we've are still in the early phase of development for cavernoma treatment with focused ultrasound. Specifically, we're still in the preclinical phase where all the research is still happening in the laboratory setting and patients have not yet been treated. And at the University of Virginia, we are exploring the non-thermal biological effect of blood-brain barrier opening, where it can be used alone or also paired with drug delivery. So let's first talk about what is the blood-brain barrier, or the BBB. So within the brain, we have all these different blood vessels. The brain is actually very highly vascularized compared to other organs in the body. And here we're going to zoom in on one of them. So the vessels in the brain are actually pretty unique compared to the blood vessels in the rest of the body. And that's because these cells that make up the blood vessel wall in the brain are actually preventing most things in the bloodstream from crossing into the brain tissue. And that's typically done to protect the brain. And it will only allow very small molecules to get across and things that it needs to survive, like oxygen and sugar. But this, of course, can be problematic when we need to treat a brain disorder because we might have a really good drug, but it can't get into the brain to be effective. So in these cases, it could be really helpful if we can temporarily open the blood-brain barrier so that we can let that drug cross into the brain tissue. So now let's talk about how we can actually do this and open up the blood-brain barrier with focused ultrasound. So just as a reminder, these sound waves are pressure waves. So with blood-brain barrier opening, this approach, we use focused ultrasound in combination with these things called microbubbles. And these are just very small bubbles that are filled with gas. And they're actually originally used as ultrasound contrast agents and still are to this day to help monitor blood flow and visualize that with ultrasound imaging. But at high pressure, the gas inside of these microbubbles will compress, causing these microbubbles to shrink in size. And then at low pressure, the microbubbles will grow in size because that gas inside of them is expanding. And so over time, these microbubbles are basically just oscillating between a large state and a small state. And so to see how this comes together within the body, we're going to zoom in here on just one blood vessel. And we'll start by injecting the microbubbles into the bloodstream, so injecting them IV. And then we'll apply our focused ultrasound to a specific region of the brain, just specifically where we want to target with the focused ultrasound. So in this case, that would be the cavernoma that we want to target. And then these microbubbles will begin to oscillate wherever they encounter the ultrasound waves. So specifically just where we're targeting with ultrasound and these microbubbles are experiencing the ultrasound waves, that's when the bubbles will start to oscillate. And this oscillation will cause those microbubbles to push and pull on the blood vessel wall, which can cause a temporary disruption of the blood-brain barrier in the sonicated region. So now if we were to inject a therapeutic agent also into the bloodstream, it can now cross this blood-brain barrier and accumulate specifically just where we targeted with focused ultrasound. And then I also just want to emphasize that this is a very temporary effect. So we typically have closure of the blood-brain barrier about 4 to 6 hours after focused ultrasound. We also expect a complete closure by 24 hours after focused ultrasound. And so you might be wondering why we'd want to use this approach for treating cavernomas. So hopefully you've seen throughout the years that there's lots of potential drugs that are being explored for the treatment of cavernomas. But this approach can be really helpful in allowing us to increase the concentration of these drugs inside of the cavernoma, essentially enhancing the amount of the drug that goes to the cavernoma rather than the rest of the body. But it can also help us to unlock the delivery of larger molecules of things such as antibodies and gene therapies, which currently haven't been explored much due to their larger size, which doesn't let them cross the blood-brain barrier very efficiently. And this table here, I don't want you to get too bogged down in, but it's highlighting many of the drugs that are being explored for the treatment of cavernomas. And I think it's really important to note that the large majority of these drugs are small molecules. So again, this is really highlighting the fact that one exciting potential of focused ultrasound blood-brain barrier opening is that it's able to unlock whole new classes of drugs for the treatment of cavernomas. And so temporary opening of the blood-brain barrier would enable candidate drugs to expand beyond just these small molecules and would make larger drug options much more feasible treatment options for CCM. And then beyond just drug delivery, focused ultrasound can also induce other effects for brain disorders. So rather than just opening this blood-brain barrier for getting things inside of the blood vessel out into the brain tissue, it can also help us to clear things out of the brain, like toxins that are building up, as is the case in Alzheimer's disease. Others have also shown that after focused ultrasound, there's a growth of new neurons or neurogenesis that occurs. And then additionally, we're providing a lot of mechanical stimulation to the blood vessel wall, which can also change the cell signaling that's happening there, which could also be potentially beneficial. And then these secondary bioeffects that I'm talking about here for focused ultrasound have been really well studied in the case of Alzheimer's disease. So here I'm going to show you where other groups have demonstrated that focused ultrasound blood-brain barrier opening can result in a reduction of amyloid beta plaques, which of course contributes to Alzheimer's disease. It can also cause a restoration of memory, the activation of specific brain cells, as well as the formation of new neurons or neurogenesis in these animal models of Alzheimer's disease. And then to provide a little more clinical perspective here, so far patients have undergone over 12,000 different brain treatments with focused ultrasound as of last year. So many of these have used the thermal effects of focused ultrasound to treat movement disorders or brain tumors. But there's also been more and more patients that are undergoing the blood-brain barrier opening regimen as well. And there's actually currently 25 different clinical trials that are either active or completed using focused ultrasound blood-brain barrier opening. The large majority are for brain tumors, but there's also a smaller subset going towards Alzheimer's disease and Parkinson's disease. So that's kind of a primer for everything. And now we're going to transition into some of the data that we have been generating in the Price Lab. So first, we're going to start out with blood-brain barrier opening and how it can be therapeutic on its own without a therapeutic drug being paired with it. So in this study, we first asked, is focused ultrasound going to effectively open the blood-brain barrier within the CCM microenvironment? So this is the first time we've explored focused ultrasound for CCM. So we need to know, will it actually work for this disease? And then we wanted to make sure that this was going to be a safe approach, especially considering that CCMs tend to bleed. And then finally, we wanted to see if focused ultrasound blood-brain barrier opening would actually be therapeutic for CCM in the absence of any drug delivery, as is similar to what we're seeing with Alzheimer's disease in this field. So in answering that first question, we started by first acquiring MRIs of our CCM mice. And then we used these images to select our cavernomas that we wanted to target. And then we began to inject an MRI contrast agent, which typically cannot get into the brain except for where there's leaky vasculature. So that can tell us if we are successfully opening the blood-brain barrier. Then we performed our standard blood-brain barrier opening approach, where we're testing a range of different pressures. And as a reminder, that pressure that we're changing there is the amplitude of the sound wave. So if we have a higher pressure, in theory, we could get more blood-brain barrier opening than if we had a lower pressure or a lower amplitude, because those microbubbles will expand more or less based off of that pressure or amplitude. And then we can confirm if we are successfully opening the blood-brain barrier by taking a post image and seeing if we're getting that MRI contrast agent to accumulate where we targeted with focused ultrasound. And if we are successful, we'll see this white signal occur from that contrast agent accumulating there. And so to walk you through these initial results of these experiments, we have in the top row here our MRI images that we initially used to select our cavernomas for targeting. This is shown by the different colored arrows, and these colors are corresponding to different pressures that we're applying. And then in the middle row, we have our MRI images taken on the day of focused ultrasound treatment, where we already injected that MRI contrast agent. So the little bits of white signal that you can see here are the baseline leakiness of these cavernomas to that MRI contrast agent. And then in the last row, we have our post-focused ultrasound images, where we can now visualize if we are successful with opening the blood-brain barrier. And we do see that we are successful because there's this increase in that white signal or the MRI contrast agent accumulating there. And we see, in fact, that there's an increase in this MRI contrast agent delivery for all pressures that are greater than 0.2 megapascals. So for all pressures that are greater than 0.2. And so next we investigated if focused ultrasound blood-brain barrier opening is safe for these hemorrhage-prone CCMs. So in these studies, we're using a T2 space MRI image that allows us to measure the volume changes of these cavernomas. And then we're using susceptibility-weighted imaging, which allows us to identify bleeding or hemorrhage. And then we're comparing these measurements in the sonicated lesions before and after focused ultrasound treatment. And so we found that there were no increases in lesion size with our T2 weighted image, as well as no increases in bleeding 24 hours after focused ultrasound blood-brain barrier opening for any of the pressures that we examined. So that's quantified here below with the absolute volume changes, as well as the change in volume for these two different measurements. So overall, this is indicating that focused ultrasound blood-brain barrier opening does not induce any acute growth or bleeding in CCMs acutely 24 hours after treatment. And then finally, we investigated if there were any therapeutic effects of focused ultrasound blood-brain barrier opening for CCM in itself. So in these studies, we're dividing the mice up into single treatment arms where they're getting one focused ultrasound treatment, or a repeat treatment arm where they're getting multiple focused ultrasound treatments staged 3 days apart. And then in both cases, we'll continue to image these mice weekly up to 1 month after treatment. So we also compared the effect of 6 peak negative pressure, which is when we're applying the same pressure over the whole length of the sonication, which is the case for all of the data that I've shown you up to this point. But we also wanted to include what's known as passive cavitation detection, or PCD, modulated peak negative pressure. So in this case, we're using the activity of those microbubbles during the sonication to actually modulate the applied pressure during the sonication. And so that's really important for us to include because this approach is what's actually being used in clinic or in the clinic or clinical trials for blood-brain barrier opening with patients to just further ensure the safety of this treatment. That allows for real-time feedback during the sonication. So at first we looked at CCM growth in sonicated or non-sonicated contralateral lesions. And as a reminder, we had previously characterized our mouse model before this study. So we knew that these lesions are tending to grow in size over a monthly period. So we expected our CCMs or our cavernomas to grow during this month period unless our sonication was doing something differently to them. So looking at the single focused ultrasound treatment arm here and the 6 peak negative pressure group, we have representative MRIs here on the left, where on the top row, we have our non-sonicated control lesions over time. And then the bottom 2 rows are our sonicated lesions with 2 different pressures being applied. And we find that our non-sonicated lesions here in black are tending to grow in size over this month period of time, but our sonicated lesions are actually stable. They're not growing during this month period. On the right, we have individual cavernomas broken down as opposed to the average, which is what's shown on the left. And we're seeing that there's not really a difference that we can tell between the pressures that we're applying. They both seem to be working well in this group. Now we're going to switch to the repeat focused ultrasound treatment arm and the 6 peak negative pressure group. And again, we find that our non-sonicated lesions are growing in size as we would expect in this mouse model, but the sonicated lesions are stable, they're not growing. And again, the peak negative pressure doesn't seem to be changing the effect here. They all seem to be performing quite well. And now if we look at our repeat focused ultrasound group with the PCD modulated approach, where again, we're using the microbubble activity to modulate the pressure that's being applied to further ensure safety during this treatment, we can find that the non-sonicated lesions grow, but our sonicated lesions are stable 1 month after treatment. So this is indicating that both the single and the focused ultrasound treatment arms, as well as the 6 peak negative pressure and this PCD modulated approach, are all capable of controlling the growth of sonicated lesions. So we're having this therapeutic effect in the absence of any drug delivery of focused ultrasound blood-brain barrier opening, stabilizing existing lesions that are treated with this approach. So next, we're still curious if blood-brain barrier opening was impacting anything else. So we started to look at the formation of CCMs. and seeing if the rate of CCM formation was altered with blood-brain barrier opening. And one thing that's important to note here is that the focused ultrasound column is actually a bit large on a mouse brain. So this is about the size of a grain of rice, which is about the top to bottom length of a mouse brain. So when we target one cavernoma, we're actually treating more than just that cavernoma. We're treating the space above and below it as well. So in this case, we're looking to see if the blood-brain barrier opening is doing anything to that healthy brain tissue that didn't have a cavernoma. So we start by first drawing on the focal column before focused ultrasound treatment, and then also drawing that column onto the 1 month post-focused ultrasound treatment. And then we mirror that column onto the contralateral side, which did not receive any focused ultrasound treatment. And then we're comparing the number of lesions before focused ultrasound and 1 month after focused ultrasound between these two conditions, so sonicated and non-sonicated. And that allows us to identify the rate of formation of new lesions in each of these groups. So here we have the results, and on the left we have in the top row T1 contrast enhanced images, which allow us to see where the blood-brain barrier was open. So where that white signal is, that's where we actually opened the blood-brain barrier on the day of treatment. And then these bottom two rows are showing us the ability to kind of look through the focal spot so that we can see the number of cavernomas in each of these conditions before and 1 month after focused ultrasound. And we see in the single focused ultrasound treatment arm with fixed peak negative pressure that there's this trend of reduced lesion formation with focused ultrasound. So one that's really important because it's showing us that blood-brain barrier opening is not causing the formation of new lesions. So we actually see that there's a trend towards a reduction of lesions forming with focused ultrasound. When we look at the fixed peak negative pressure and the repeat focused ultrasound group, we actually see now the statistically significant reduction in new lesion formation. And then finally, if we look at the PCD modulated approach, we again see this mild trend towards a reduced number of lesions forming. And again, there's not an increase with this approach. So ultimately, the data is indicating that not only does focused ultrasound have this protective role of controlling the growth of sonicated lesions in the absence of therapeutic delivery, but it also has this potential to have a preventative role for CCM too. So if we can figure out the right dosing and timing of focused ultrasound, we might actually be able to prevent new lesions from forming. And then this would also be really beneficial, of course, for patients that have the familial form of CCM. This is what our mouse model is basically, the familial form of CCM. And this approach will also have this ability to still seamlessly integrate with therapeutic delivery to further enhance the potential for CCM treatment. And that's what I'm going to basically talk about in the next section. So to wrap up this part of the talk, we demonstrated that we could apply focused ultrasound for CCM, and in fact, this was the first case of that being done with focused ultrasound for CCM. We saw that focused ultrasound can open the blood-brain barrier and enhance MRI contrast agent accumulation within the CCM microenvironment. We also see that focused ultrasound blood-brain barrier opening does not induce an acute growth or bleeding of CCMs despite them being prone to bleeding. We also find that blood-brain barrier opening does control the growth of sonicated cavernomas, and that repeat blood-brain barrier opening with a fixed peak negative pressure approach can be useful for reducing the formation of new lesions. So that was all about blood-brain barrier opening being therapeutic without any therapeutic delivery, so without any drug delivery. But now we'll talk about how this can be further used to enhance this effect by also pairing it with drug delivery, and if that's going to be possible. So in order to do this, we use what are known as model drugs. So basically, we're not actually injecting therapeutic drugs into the mouse, but we are going to use these MRI visible drugs that we can get as proxy for therapeutic delivery. So we know the size of these molecules. They're visible to the MRI, so we can track them and monitor them and where they go and how much goes there. And then we can use that as a proxy for how much of a drug of that size would also be getting to the cavernoma. So To do this study, we leveraged a kind of fancy form of MRI known as T1 mapping, and that's allowing us to measure the permeability of cavernomas and blood-brain barrier opening in a more quantitative way than we were doing in this last section that I showed you. And we're also using the sequence to look at the delivery of model drugs. So this is going to be multi-HANCE, which is a very small molecule. It's about 1 nanometer in diameter. And we'll also look at what's known as gadospin, which is this other MRI visible drug that's a little bit larger. It's about 5 nanometers in diameter. And gadospin is a bit more similar to something like a small biologic, like a protein that might be used to be delivered and be therapeutic. So first, we wanted to see if focused ultrasound blood-brain barrier opening could increase the rate of the small molecule delivery to CCM. And so for us to do this study, we first got a baseline by acquiring the MRI before any contrast agent or model drug was injected. And then we acquired this MRI post-model drug being injected. We imaged for a little bit, and then we applied focused ultrasound to just one hemisphere of the mouse, leaving the other hemisphere not sonicated. And then we again acquired our MRI post-focused ultrasound and contrast agent delivery. And then we were able to measure the contrast delivery in the time period before focused ultrasound and the time period after focused ultrasound in this focused ultrasound treated and non-focused ultrasound treated hemisphere. And in the slides going forward, we're going to break this down a little bit further. So we'll also look at what I'm calling the interlesional space, so inside the core of the CCM, as well as inside the perilesional space, so the surrounding cavernoma space, which is kind of like the space that therapeutically might be useful to treat because that's where the CCM is expanding into. So we'll look at that for both the sonicated and non-sonicated condition as well. So here we have the results from this experiment. So on the left is a reference MRI image, and then we have on the right our concentration maps where we can actually quantify the concentration of these model drugs, with yellow being a higher concentration of the drug and blue being a lower concentration of the drug. So on the top is 10 minutes post contrast agent or model drug injection without focused ultrasound. And then on the bottom is 30 minutes post-injection and about 10 minutes post-focused ultrasound treatment, where we treated just the right hemisphere in this case. And you can see that there's this increase in concentration of this drug with the more yellow signal here. And then quantifying these concentration maps through the CCM core, or the interlesional space, we find that focused ultrasound is significantly increasing the rate of the multi-HANCE, or the small drug delivery. And in fact, if we compare the slopes on the last half of this timeline to the first half of this time graph, we can see that focused ultrasound is actually tripling the slope, or the rate of contrast delivery in this case, the small molecule delivery, compared to the side that was not sonicated. And in fact, this slope compared in the last half to the first half for the non-sonicated condition is actually decreasing. So it went down 1.5 times in this extra 20 minutes after focused ultrasound when it wasn't treated. And then if we look at the space surrounding the CCM and the perilesional space, we also see that focused ultrasound is increasing this rate of small molecule delivery, this time 5-fold. And again, we see that the non-sonicated side is actually decreasing the rate of delivery over the same time period. So ultimately, this data is indicating that focused ultrasound blood-brain barrier opening is increasing the delivery rate for small molecules. And so next, we wanted to see how much focused ultrasound blood-brain barrier opening could enhance the total amount of the model drug making it to the CCM. So that's more clinically meaningful. Here, we're just looking at the rate of the delivery. And now we're going to look at how much of the drug is actually getting into the CCM. How much could we expect a therapeutic to make it into the CCM? So for these studies, we start by injecting either model drug, so the small molecule, multi-HANCE, or the larger molecule, gadospin. And then we can elect our T1 mapping images, the fancy MRI that lets us look at permeability. And we do this just for the case without focused ultrasound. So that gives us that baseline, what would we expect of that drug to get into the CCM without focused ultrasound. And then 1 day later in these same mice, we apply a focused ultrasound blood-brain barrier opening to just one hemisphere. and we inject that contrast agent again and perform our T1 mapping. And now we can look at how much drug got in with focused ultrasound treatment. So in this case, we're able to compare the same mice and the same lesions, how much blood-brain barrier opening increases the total delivery of each model drug. So looking first at the multi-hands or the small molecule data, here we have the MRI image before injection, and below is the concentration map 20 minutes after injection. Now we have the same concentration for the same mouse, but with focused ultrasound treatment. And in this case, we again treated just the right side, and you can see that there's an increase in the molecule, the small molecule getting in with focused ultrasound. And so if we quantify this data, we see that the multi-hands concentration inside of the lesion, we see that focused ultrasound is significantly enhancing the small molecule delivery at 20 minutes post-injection. And if we integrate this data over time, we can look at the area under the curve or the exposure of the CCM to the model drug. And we again see that focused ultrasound is significantly increasing the exposure of multi-hands inside of CCMs. If we look at the perilesional space now, we also see that focused ultrasound is enhancing the delivery at 20 minutes after treatment, as well as the exposure for small molecules. And so in this case, we're able to tell that we can predict how much more of a small molecule drug would be delivered to CCMs with focused ultrasound blood-brain barrier opening treatment. So we do see that focused ultrasound is enhancing almost up to threefold how much of a small molecule would make it into and around the CCM. So it's obviously very important for hopefully increasing the therapeutic effect of these drugs, as well as minimizing side effects. So if we repeat this process for the larger molecule Gaddiston, which is more similar to a biologic drug, we again see that focused ultrasound is significantly enhancing the delivery and the exposure of the larger molecule inside of the lesion. We also find in the area around this lesion that the exposure and delivery are also enhanced with focused ultrasound. And so again, the study is helping us to predict how much more of a biologic drug would be delivered to CCMs with focused ultrasound. And we can see that without focused ultrasound, the delivery is essentially zero for this size molecule. So by using focused ultrasound, we're actually enabling new classes of drugs to be used for CCM beyond just small molecules, because it doesn't seem like that's really going to be a feasible option, at least with the type of molecule that we're using in this case. We don't see much delivery getting in without focused ultrasound. So finally, we just wanted to compare the delivery enhancement effect of focused ultrasound for these two molecules. So first, we looked inside of the lesion, and we find that there's this trend of Gaddiston having a larger increase in exposure than multi-hands, so the larger molecule having a greater exposure inside the lesion than the small molecule, suggesting that there's a benefit of focused ultrasound delivery that's greater for larger molecules in the CCM core. If we look at the space around this lesion, we see that these two are pretty much the same. And so if we next compare the location that's most enhanced with blood-brain barrier opening for these two contrast agents and examine the ratio of the intralesional and the perilesional, we see that the small molecule tends to be a ratio less than 1, which is indicating a preferential enhancement in the space around the lesion. Whereas the larger molecule, we see that this ratio is mostly greater than 1, which indicates that there's a preferential enhancement with focused ultrasound in the CCM core. So collectively, this is indicating that the focused ultrasound blood-brain barrier opening is affecting these different size drugs differently in the location that they're most benefiting from. And so just to wrap this part up, we saw that focused ultrasound can enhance the delivery rate of small molecules to CCMs. We see that focused ultrasound enhances small molecule and biologic size molecule total delivery to CCMs. And then we see that focused ultrasound is differentially enhancing the delivery of small molecules with a preferential treatment going towards the perilesional space and biologic model drugs to the CCM core. And so looking forward, we are currently in this preclinical space, but we are very excited to hopefully move this into clinical trials and get FDA approval and US and beyond reimbursement. And as a small step towards that, we at UVA have been able to actually take a glance at what this might look like, what clinical treatment of CCM with focused ultrasound might look like. So thanks to Doctors Jason Sheehan and David Schlesinger, we were able to reimagine the treatment plans for CCM patients that received radiosurgery. And here I'm just going to show you an example of one patient. But these patients received radiosurgery because they were not candidates for conventional surgical resection. And unfortunately, about 11% of the patients that received radiosurgery from this study experienced adverse radiation effects. So hopefully we can minimize those effects with focused ultrasound, and using the NAPIFES clinical system, which is one of many clinical systems that are available for focused ultrasound at this point. Dr. David Moore at UVA designed some focused ultrasound blood-brain barrier opening treatment plans for these same patients. And so those are shown here in red. These are many different focal spots that are able to treat this cavernoma that's about 2 centimeters in diameter. And we basically are able to tell that these CCMs are accessible for focused ultrasound treatment with the software and this technology, which can offer an option without radiation and more non-invasive options altogether. So ultimately, we can see that this technology is actually ready for these treatments. And hopefully, in a short period of time, we'll have the science there and some clinical trials ongoing as well. So going forward, we still would like to really understand therapeutically and mechanistically what is happening with focused ultrasound for CCM. Why is this therapeutic, especially by itself, and looking at the cellular response following blood-brain barrier opening, as well as how mechanical stimulation is contributing here. We want to evaluate the therapeutic potential of focused ultrasound in more models of CCM. So in this case, this was a CURT1 or a CCM1 mouse model, a familial model. So we'd like to expand that to other mutations as well as the sporadic case as well. We'd also like to look at treating CCMs in other locations. So most of these lesions were confined to the striatum, but we of course would like to look at other brain locations as well as spinal cord if possible. And then we also want to continue to look into MRI techniques to understand our focused ultrasound treatment better. So looking at later time points and understanding how long these drugs are retained in the CCM, when we might need to redose, and also looking at the delivery of even larger molecules. And so finally, I'm going to wrap up now. I just want to acknowledge everyone who has made this work possible. So I need to, of course, thank the Price Lab and my mentor, Richard Price, who guided me on all of these studies and provided a lot of the funding and support needed to actually do this work. I want to acknowledge Tanya Cruz, who's continuing to take on this work now that I've graduated. And I need to highlight Peter Tverdik and Khadija Sharifi, who generated the mouse model that we used in all of these studies, as well as Wilson Miller, who also optimized the MRI that we needed to use in this study. Of course, none of it would be possible without funding that we've received from the NIH, the Focused Ultrasound Foundation, the Alliance to Cure. And I also just want to put in a quick plug for the Focused Ultrasound Foundation. If you want to learn more generally about focused ultrasound, they have a really good website, a lot of good information on there. So that's written here, bestfoundation.org. And with that, I think I'm done for this part, and we can turn it over for some questions.
Thank you. Thanks, Delaney. That was wonderful. Can you stop share for a bit, and then we can see each other better? Thank you. There you go. All right. So we have 20 questions in the queue, over 20 questions, but about 35% of them are the same question. Everyone wants to know about brainstem lesions and the potential of focused ultrasound for brainstem lesions. Is there any reason at this moment to think that it won't one day be applicable for them?
No, I think brainstem lesions are going to be one of the best candidates for focused ultrasound. Obviously, those patients would be the ones that are less likely to receive surgical resection or be looking for alternatives to surgical resection. So of course, we want non-invasive treatments for them. Brainstem lesions should be targetable with focused ultrasound. A lot of times it's actually easier to treat deeper targets than more superficial targets with focused ultrasound because of the way the transducer is designed for human patients. Is you kind of have this helmet that you wear with lots of different transducers, which are the elements that actually produce the sound energy. those all kind of have to converge together. So deeper targets can be better than really superficial targets because it's hard to get all of those to actually converge. I also want to just say that Natasha Shea Bonney's group, who's also at the University of Virginia, she has a student, Steph, who is working on cerebellum cancer, like tumors that she's targeting with focused ultrasound. We heavily collaborate with them. She was actually a student of the Price Lab when I started. And so hopefully with Steph's protocol, we can also begin to expand looking at CCMs in the brainstem area as well. So that's all going to help synergize together. There's a lot of good projects that help us all learn and grow both in the cancer space, also in the CCM space.
And I want to break in and say that we are already talking with the University of Virginia. The Alliance is talking with the University of Virginia about starting a clinical safety trial. We're also talking with the manufacturer of the NaviFest system. There is a unit of that system at the University of Virginia, and there will be units installed at Stanford and at Brigham and Women's in Boston within this calendar year. I think if fingers crossed, all goes right, and we have all of our ducks in a row, there's a potential that sometime next year we'll have the first CCM patient have focused ultrasound treatment for their cavernous malformation. It will probably not be someone who has an inoperable brainstem lesion because this is a safety trial. So we're looking for people who, well, we'll find out who we're looking for, but I can tell you that is probably not the first person we're going to be looking for. We're still trying to figure that part out. Okay, so the next question is the same, along a similar line, spinal lesions, spinal cord lesions. Will that be an option?
Yeah, so that should also be an option. Personally, our lab has not had a lot of experience with spinal cord treatment with focused ultrasound. That said, there are other groups that are specifically using focused ultrasound blood-brain barrier opening for spinal cord. And as that begins to take off more and more in the clinical space as well, that should be more than able to cross over for CCM as well. So that should technically not be a problem. There's no reason to believe that that shouldn't be another area. I think it's just unfortunately a little bit slower because that hasn't been the main focus of focused ultrasound over the years. It's been primarily brain focus, and then it's starting to expand into other CNS areas, including the spinal cord.
Okay, and then the next question is, what about someone who has hundreds of lesions? What could focused ultrasound do for them? Yeah,
so there's no reason to think that we couldn't treat really large volumes. There's actually a group in Australia, Jürgen Goetz's group, who actually uses what's known as scanning ultrasound. So they basically take the transducer in mice, and they're just moving the focused ultrasound transducer to cover the entire brain of the mouse. They haven't seen adverse effects for that, and they're looking into it for Alzheimer's disease. They're the main group that has shown that focused ultrasound blood-brain barrier opening without therapeutic delivery has been beneficial for Alzheimer's disease. So we've looked at a lot of their work to inform us for CCM. So I think a similar approach will also be possible. Clinically speaking, most of those systems have not been designed to scan, unfortunately, but I think it's doable. It might just be that we're treating one chunk of the brain in one spot, then we'll move on. It won't be as seamless perhaps, but it should be possible. So that is the nice advantage, is that it's non-invasive, you know, it should be much more feasible to treat a large volume than it would be surgically.
Okay, and then the next questions are just about the actual procedure, which is, does focused ultrasound work through the skull, or do you need to make an opening, and is it painful? Those are our two questions we've gotten a lot.
Yeah, so there's many different clinical focused ultrasound systems currently available, and they all work a little bit differently. The NaviFest system, which is what we've kind of highlighted more tonight, is kind of this like half helmet, which works transcranially, so which means that you're not opening the brain or doing anything like that. You just kind of hold it and couple it, usually with a water balloon. I don't have a great word for that, but this kind of like water coupling of the device to the brain. And so that this technology is kind of like maybe a second generation clinical technology. So it's come back from the days of the first generation, which was actually this helmet that covers the entire head. in that case, it still wasn't invasive in that you didn't cut open the skull at all, but you did have to wear this kind of device that lets you screw the helmet onto the head. So you would have a minor incision into the flesh just to secure that helmet. So NaviFest should be more non-invasive. It's more distal. You're not drilling in. That being said, there are clinical systems for people that are receiving repeat doses for cancer, where they actually implant the focused ultrasound device. They take out a chunk of the skull, put that there instead. That's just to make it more feasible, because these procedures can be a little bit hard to schedule in that you have to get an MRI suite, you have to have the neurosurgeons, the nurses. So doing repeat doses can sometimes be logistically harder. So that was the solution there to get around it. If we implant it, we set it up the first time, and now you can get as many repeat doses pretty conveniently. So there's a range of options. What we're talking about here with NaviFest is quite non-invasive, though. There's no scalpel, there's no incisions.
And so you're talking about repeat treatments. Do you think that repeat treatments would be required for CCM?
We don't know yet. We see some beneficial effects even in our single focus ultrasound treatment arm. That's looking out to 1 month in mice. We obviously have a big question here of how the human translation is going to go. These initial safety studies will hopefully give us a lot more insight. Unfortunately, there's not models of CCM for larger animals that really help us kind of bridge that gap. But I think it's possible that repeat treatments will be more effective. It might just be a question of, is it necessary, which will probably depend on every person a little bit differently, because everyone's cavernoma really does seem to be very unique. We hope, though, that the technology continues to advance, which we know that it is. This field has been really rapidly growing. It's changed a lot since just the 5 years that I've been introduced to it throughout graduate school. So I think that as the field grows, we're getting all these better and better, more accessible focused ultrasound clinical systems, which makes all of these logistical problems or, just technical issues a lot more minimized. So I think the repeat regimen will hopefully be more and more convenient as time goes on as well.
Then there were questions about the lesions themselves. The size, the smallest lesion that can be sonicated, the largest lesion that can be sonicated, and also can lesions that have hemorrhaged be sonicated? Mm-hmm.
Yeah, so in terms of smallest lesions, I think even tiny lesions can be targeted as long as they're visible with MRI, where our results are showing that blood-brain barrier opening is still safe in the tissue that is not a cavernoma. So I think even if we're targeting larger than the cavernoma itself, that might actually be helpful, but we're seeing that is at least not harmful. In terms of the largest lesions that can be treated, the nice thing with focused ultrasound is that we can continue to stack multiple focal spots. So we don't have to get everything in just one focal column. We can just keep putting more and more focal spots together so that we're able to cover that entire cavernoma, even if it is quite large. It might just be a longer treatment. Here we're sonicating, like one focal spot is being sonicated for a 2-minute duration. Obviously, they're still set up to go into this initially, getting the MRI, getting the mouse position. A similar phenomenon happens for human patients, but stacking on 2-minute focused ultrasound sonications really is not going to be too much longer of a process. Hemorrhaging CCMs. So we do, in our mouse model, we know that these lesions are hemorrhaging over time. We didn't necessarily screen for a lesion to have hemorrhage previously or to have not hemorrhaged previously for selecting these cavernomas, but the odds are over the course of these studies, we treated a lot of lesions, I think close to 50, if not more. So odds are is that we treated hemorrhaging lesions during this time period, and 97% of our lesions were stabilized in growth with the focused ultrasound treatment. So it seems like hemorrhaging or non-hemorrhaging lesions are acting similarly. That being said, we do kind of need to break that down more rigorously than we have currently done to understand better.
I know we have about 3 minutes left, but here's a somewhat complicated question. Folks are asking you just to quickly differentiate between stereotactic radiosurgery, gamma knife, focused ultrasound, and lit laser ablation, and how those are different from each other.
Yeah. Okay. So gamma knife or stereotactic radiosurgery involves, it's non-invasive, generally that you're not opening the skull in any way, but there is ionizing radiation that is being involved to treat, and basically you're causing DNA damage to kill those cells. With focused ultrasound, that's just sound waves. It's similarly completely non-invasive, but it's sound energy. It's non-ionizing radiation. With lit or laser, with but that is a laser, so a light form of energy that's traveling into the tissue. With light, the penetration of light energy is less than the penetration of sound energy, so you tend to need to remove a chunk of the skull because that gets the light gets absorbed by the skull. You need to expose the brain to actually perform the lit procedure. So those are the ranges. You kind of have radiation, but non-invasive, non-radiation, non-invasive with focused ultrasound, and then slightly invasive, minimally invasive, and light energy.
That was good. That was really quick. Still 2 minutes, so I can ask you at least 1 or 2 more questions. Looks like the post-focused ultrasound scans are at 30 days. Did you follow up with these mice at any longer intervals?
So we did not. Unfortunately, our mouse model, we're kind of pushing the lifespan of this mouse model. So we began treatments between 2 to 3 months of age for this mouse so that they would have a high enough lesion burden for us to treat multiple cavernomas. We would follow them up to that 4 month or 4 to 5 month time period, and then these mice tend to pass away around 5 months old. So we needed to end the study, unfortunately, to collect that. But that being said, that is the lifespan for this mouse model, which kind of bodes well for this disease being quite highly established. It was an early time point of the disease.
Okay, 1 more question, and I'll make 1 comment. 1 comment, a lot of you are asking about Australia. Yes, Navifus has machines in Australia and in Taiwan. Those are the only 2 other places currently besides the US. The question is, so if you're delivering medicine, small molecules to a CCM lesion, how do you know that the level of the drug that you're delivering is adequate for treatment? Can you titrate the dosage at all using the focused ultrasound?
Yeah, that's an interesting question. So systemically, you can control the dose. So what actually goes into the bloodstream can still be changed. In terms of modulating the focused ultrasound to titrate the drug, that's less well understood and probably will be different for each drug. So that would probably have to be really routinely looked at for each clinical trial pairing with a different drug, and will be, I'm sure. But you could potentially play around with the focused ultrasound parameters, so the pressure that's being applied, how long the focused ultrasound is being applied, if you're doing the repeat dosing to get more or less of that drug in. But again, that will all have to be screened in this clinical trial to understand better.
Excellent. Okay, so the video should be on our YouTube channel in the next week or 2. We have more questions in the queue, but I think we really did cover the bulk of them, so thank you, thank you, thank you. If there are any other questions, and you know what, I'm going to drop in on the support group and hang out there for a little bit and see if there's any questions that folks have that I might be able to give an answer to, or write down to send over to Delaney when we're all done. Thank you, Dr. Fisher. Thank you.
Yeah, it was great to be here tonight. I appreciate the invitation.
Yeah, and thank you for the work. We're all very, very grateful. All right, good night, everyone. The link for the support group is in the chat. Please go ahead and click on it so that you can get there and join us, and I will be there in just a few minutes. I might not join right at the beginning, but I'll join soon after. Thank you. Good night.
Thanks, everyone.